Hello, everyone. It's an honor to be invited on this wonderful microTAStic series. Today, I want to share a talk about investigating intratumor heterogeneity of cancer metastasis using an IDRD area. And my name is Xuejie Zhang, and now I'm starting my PhD career at City University of Hong Kong under the supervision of Professor Chen Huang. Uh, and sorry, and as we all know, currently the microfluidic-based platform have allowed us to study cell behaviors, functions, and cancer related mechanism at single cell and resolution, and this kind of resolution has generated significant leap for today's research. However, for current microfluidic-based platform, faced one biggest obstacle is how to achieve long-term single-cell culture with very high throughput. Therefore, we proposed our device to try to achieve this kind of purpose. Through comparing with other well-known and well-established platforms such as microwave printed droplets and the Pico injector, our proposed device could achieve high efficiency no matter it's at throughput or the long-term single-cell culture. And here is the design of our DLD device. Actually, is a device based on the DLD area, but it contains three different phases. The first one is the water in oil droplets. The second, the middle, in the middle phase is the emulsifier as the droplet goes through this area. The surfactant at the surface of droplet will be gradually washed out, and finally, the droplet will be broken out and release the contained cells into the fresh medium, and then the dirty cells will be washed and cleaned up and finally be re-encapsulated into the oil directly. As you can see in figure B, firstly, we use the different color labeled phases to show that the laminar flow could be stably formed in our device. And in the figure C, there is a short video to show how the droplet moved inside our device and how it gradually be broken out and finally merged with the fresh media. And through the process, as we can see, the force generated by the droplet broken did not interface the interface between oil and the aqueous phase. And because just like I said, as we all know, the interface between water and the oil actually is very stable. Any inappropriate operation could disturb and cut out the aqueous phase. Therefore, in order to sustain this stable laminar flow for several hours, we designed a pillow area at the outlet of the oil. And this, as we can see, the surface tension generated by the pillows significantly help to stabilize the interface between oil and the aqueous phase, so that the cell will be better exchanged with fresh media. And even when we label the cell with level and death dye, we could also see a very clear trace of the cell movement. And then in order to show the exchange efficiency of our IDRD device, we labeled the droplet and the fresh media with CY5, the red fluorescent dye, and the green fluorescent dye separately. As you can see in the figure, through our device, actually, we could achieve nearly 100% medium exchange efficiency. and then we applied our device for the long-term single cell culture, and we could achieve maximum to 5 days 5 day culture, and the cell could sustain the viability up to 19%. These two experiments, we showed the exchange efficiency achieved by our device and provide opportunity for the long-term single cell culture without causing the too many cell deaths. And because we use the fresh medium to wash out the cells, therefore it will cause a lot of empty droplets. These empty droplets will interfere with our later study. Therefore, in order to enrich the droplet with cells, we applied our well-established and well-applied AI-powered droplet sorter to enrich the cell content droplets, and this platform could be very well performed in our lab, and we also achieved a very highly high concentrated droplet with cells. And for our next step, we hope to use also this well-established living surface capture by our teammates to capture multiple targets such as cytokines, chemicals, or even exome, because as we all know, this kind of secretions play very significant role for the cancer functions, cancer drug resistance, and especially the cancer metastasis. And we hope in future this platform could capture different targets at different time points through continuous medium exchange with our device. We hope this device will provide more insightful opinion about the cancer metastasis and the related mechanisms about cancer recurrence. And that's all I want to share today. Thanks for your listening.